Wow. Kia ora. Good morning, everybody. My name is Michael Lesser. It's my pleasure this morning to introduce this morning's plenary lecturer, Dr. Virginia Weiss. Could I have the first slide, please? Virginia is a distinguished professor from Oregon State University, and she has been recognized by our own institution in various societies. For her excellence as a researcher and as a mentor to graduate students and undergraduate students. Slide, please. There it is. There she is. I call Virginia a cellular ecologist. And the reason I do that is because she's fundamentally interested in the establishment, maintenance, and breakdown of symbioses, and then has the ability to take those cellular level phenomenon and scale them up to the organism and scale them up to the ecology and evolution of symbioses in general. She is the first author of a seminal paper in Trends in Ecology and Evolution with some other familiar names there, where she made a strong case for the development of organismal models to study corals, and in particular, issues related to symbiosis and at the time bleaching. So that model that she made the case for was aptasia, now known as exaptasia. Next slide, please. So Virginia and I are contemporaries. We were both in graduate school about the same time, and both postdocs at about the same time in the late 80s, early 90s. And we first met at the 1991 Edwin Pauly summer program in marine biology, where we were invited lecturers along with Mark Patterson to study diffusion barriers, carbon limitation, and reef corals. Great bunch of students. We conducted studies, multiple kinds of studies, including ones for ourselves, which resulted in this paper on the bottom, which is still, I'm happy to say, quoted in the literature. And we were all led by our mentor at the time. Paul invited us all, Dr. Paul Joe Keel. And I'd like to make a plug for Paul. I think it's long overdue. I'm going to abuse my time here. I think it's long overdue that the society recognize Paul for his incredible contributions to coral reef science, and also the training of hordes of coral reef scientists that many of you are sitting in this room right now. So that's my 2 cents, and with that, I'd like to have Virginia come up to the stage and give her a round of applause, please.
Good morning, everyone. So I really appreciate, and I'm so honored to have this opportunity to speak to you all. We're here together for an amazing set of interactions and exchanging of new information and learning new faces and meeting people. As a subset of that, excuse me, we're in the middle of 2 days of cell biology, a very large session that's in room 516. And then there's lots of it sprinkled around other sessions through the week. So my job today is to not speak to my colleagues as much. They can critique me, because I'm sure I'm going to say things that maybe aren't quite what they want to hear, or they want to hear more, or I didn't give the right detail. But for the rest of you who aren't cell biologists, I want to try to show you the beauty of coral reefs, not just at the reef level, but at the cellular level, and just how amazing these organisms are, and how we have tried to discover that those interactions through the years. So today I'm going to talk about regulation a lot of the time. And that's been the focus of my group for 30 years. And I'm going to talk about mechanisms that underlie the initiation and maintenance of the partnership, and also mechanisms underlying dysbiosis or cnidarian bleaching, so how the partnership falls apart. And to give you a roadmap of the talk, first I'll start with just a brief overview of what corals are. Some of you will know all of this, others of you may not. Corals as microbial ecosystems, the intimacy of the symbiosis, the value of model systems. Michael set me up nicely for that. And then I'm going to talk specifically about regulation, regulation of the cnidarian dinoflagellate partnership. And I'm going to tell two small stories from my lab that I had to pick out of many over the years. The role of host innate immunity in regulation, and I'll tell a story about NOx, which is an enzyme, and host symbiont biomass regulation. And I'm going to tell a story about the role of nutrition and symbiosis in host asexual development. So when we look at a reef, this was a photo taken by Ove Hergoldberg, who's speaking tonight, back in 2007 when I was visiting him on the Great Barrier Reef. And we see on a healthy reef this beautiful abundance of life. And the corals and the soft corals around them have this beautiful brownish hue. And there are 10,000 species of cnidarians and about 6,000 species of anthozoans that include corals. And many of them engage in a symbiosis with dinoflagellates and other things. So really, corals are microbial ecosystems. If we look at this bush of life, we can see that the host is here in the eukaryotes, with the opisthocons, but the red underlines are all different types of symbionts that occur within the host body. And together, all of these things are referred to as a holobiont, as the holobiont. And so if you look, for example, at the little larva up here, you can stain that. First of all, the algae themselves just autofluoresce under a fluorescence microscope, but you can also stain the bacteria, and you can see that even in a little larva that's just a tenth of a millimeter, you see both the giant algae that are inside the host, but also a lipstick of little bacteria that are surrounding the mouth of the larva. So these microbial ecosystems are incredibly dynamic. There are sessions throughout the week that are describing other parts of this interaction, the microbiome part of it, how disease is impacting reefs, all these things. But it gets down to the holobiont really being a microbial ecosystem. So then we can talk about, when we get to the cellular level, we can talk about intimacy and talking about endosymbiosis, but also about autotrophy and heterotrophy. So when we look at a coral, and we look up close at a polyp, and we look up even closer to a polyp tentacle, we can see that the inside of the tentacle is arrayed with thousands of coccoid golden spheres, which are the dinoflagellates. And they're embedded deep within the body of the animal. They're not on the outside. There's a thin layer of epidermis, and then inside, by the gastric cavity, are the gastrodermal cells that are containing the algae. So the symbiosis is intimate. We can make a cartoon of this, and we can show that the algae are actually surrounded by a membrane called a symbiosome. And we now know more and more about the symbiosome. There were some amazing talks yesterday about that by one of my former students. So we know quite a bit about how that symbiosome comes about. But it's deceptive to show a cartoon like this, because actually the cell looks like this, okay? So what you're looking at is these tiny little host cells with these bowling balls in them, right? And so it's very intimate. There's very little host cytoplasm. There are tiny little nuclei there and there, and the rest of it is kind of made up of algae. So it's pretty hard to image these things, and it's complicated. We also heard a really cool talk yesterday about showing how the algae are moving through the host. So imaging has been challenging, but we know a little bit, and we know how intimate it is. Then when we talk about nutrition, the central dogma of the symbiosis is that there's nutritional exchange between the host and the algae, whereby the host is providing the algae a safe home and inorganic nutrients, carbon dioxide, nitrogen, phosphorus. And the algae are in a high light environment doing tons of photosynthesis and transferring that carbon over to the host cell. Sounds simple, but it's pretty complicated. So you can call a coral autotrophic, so an animal that's autotrophic, but it's actually a symbiosis. But also, in most cases, the animals are also feeding, and we increasingly know how important heterotrophy can be, especially in the context of stress and bleaching. And so that makes the story even more complex. And this whole situation is highly regulated. So understanding the regulation and how that there's a give and take between nutritional status and regulating those partners is a big part of what many of us in the cell world do. We can also talk about the diversity of the symbiotic algae. Way back in the day when Michael and I were on the boat in Hawaii, there was one type of symbiont, and that was so much easier. I mean, way easier. This whole taxonomy thing, it's really overrated. But we now know that there is enormous diversity of the symbiotic algae. There are 11 genera, and there will be more described by those annoying people. But you can see that it spreads across many different types of hosts. So these are the hosts here. And so different types of symbionts occur across a large diversity of hosts. And crucially, there are many algae in this clade, Symbiodiniaceae, that are not symbiotic. And so it's really the biology of the Symbiodiniaceae is amazing, a story unto itself. The dinoflagellates break all the rules. They have giant genomes. You can see here the genome sizes are really, really large. So it's made studying them challenging. But I'll try to point a few places to where the cell biology of the symbiosis is moving towards solutions. And I'll just say here that host symbiont specificity is really diverse. So how the partners mix and match each other, it's diverse, it's complex, and it's dynamic. And there are enormous numbers of folks in the room or in the building who are really interested in finding out if managing or altering this specificity can help us with coral resilience. So can we breed or drive evolution of corals to become more resilient by fiddling with that specificity? Because some symbionts are better at heat, some hosts are better at heat, and so there are multiple sessions that are addressing that very question. So to talk a little bit about model systems, so we don't agree to call this Exaptasia diaphana by its actual name. Most of it's just still call it Aptasia, so that's what I'm going to do. But it's Exaptasia diaphana, which is a lovely aquarium pest that grows really, really well in the lab. And it has there's a ton of value in using model systems to push forward our understanding of coral biology. Aptasia is clonal, it grows in the lab well, it has both sexual and asexual reproduction in the lab, and I'll show you that later. And then you can bleach it experimentally and keep it aposymbiotic. So this lovely photo here taken by Chris and Ting Ting shows that aposymbiotic Aptasia, unlike corals, can do just fine in the lab. And so there's so much value in being able to manipulate that symbiosis and adding algae back and taking it away and all those sorts of things. The symbionts themselves are also well studied at this point. There are a variety of species that occur in Aptasia, and many of them have completed genomes. And so there are lots of Symbiodiniaceae that cannot yet be cultured, but many of the ones that occur in Aptasia can. So we have a lot of power to study how the symbiosis works. And this has been something I've been championing for a very long time, as Michael mentioned. And this is just, if you're not on this map, you can come and I'll put your dot on later. But these are the labs that I can think of, or the places in the world that are really studying Aptasia. And the point is that we're interactive and we trade cultures and we share information rapidly, and that helps us accelerate the pace of discovery. And that's one of the main values of models, one of the main values. This is a photo from back in 2007 on Heron Island where Simon and Ove and John Pringle and I organized a workshop, and we brought together non-coral biologists, people who studied model organisms, parasitologists. They were pretty psyched to get to go to a reef. Together with reef biologists, and we talked about the value of models, and really that's when the Aptasia world really took off. So it's been a long-term goal of mine, and I'm very proud of that. And just as a little side note, just an example of solutions-based work that I'm involved in, I'm working with a group to develop cryopreservation tools for Aptasia. There's a lot of interest in cryopreservation of coral material, and we can have Aptasia spawning once a month, and so that's way faster than once a year. And so we're really actively involved in trying to push these tools forward in Aptasia so that we can not only have these tools available for Aptasia community, but also to help with coral cryopreservation goals. Okay, so the Weiss Lab through the years, it takes a village. This is just a photo of some of my group through the years, and the only thing that changes is the color of my hair. And this photo was featured by Shimpei yesterday, which was a little shocking, but because he stole my thunder. But at any rate, this was the very first photo back in 1998, 2 years after I started my lab, and then up to today. And the point of showing this is that, you know, I'm so proud of the people I've been able to train. I'm so proud of you for the work that you're doing. Many of you are here. And it's that passing of knowledge and taking a mutual love of discovery that makes us all excited to be here and all excited to work for solutions for coral decline. So let's get to the science part of the talk. We can ask big questions in the regulation of symbiosis, and these questions have actually been around for like 50 years. Some of them are now getting to the point where we feel like we're solving them, which is really exciting. But I remember years ago when I was a young assistant professor, I thought of all these really cool ideas and how original they were, and then I went back and read the work on green hydra from the 1980s. I'm like, oh my gosh, these are the same questions. So these are hard questions to answer, and that's why it's taking us a long time, but we're getting there. So we're going to start and we have two areas that we're going to talk about today. The first is immunity, innate immunity. And we can ask questions like, what cellular signals are involved in host symbiont recognition? Does the host mount an immune response that is modulated by the symbiont? So how do the symbionts get in and stay in? How does that happen? And how does signaling and immunity change during symbiosis dysfunction and collapse? So let's dissect this immunity thing a little bit. And we can do it by learning a little bit about how 85% of corals start off symbiosis. So about 15% of species actually vertically transmit their algae into the egg, but the majority, 85% of species, actually have to, have to acquire symbionts anew with each generation. And you can study that if you can be present for a coral spawn, and you can have algae to add to the corals. Okay, yes, that's good enough, thank you. There we go. Okay, so here we have photos of, this was work done by my very first graduate student, Jody Schwartz. You can have a larva, this was Fungus Scuteria from Hawaii, that spawns predictably in the summer times. You can take algae from an adult and add a little sousance of brine shrimp, like make it a little bit hungry, and it spews out this thread of mucus, and it trails it behind it as it swims, and it captures algae in the mucus here, and then kind of brings it into the mouth, kind of like going down the toilet, and sure enough, over time, you'll have a symbiotic larva. And you can really study this in some detail, and with Aptasia, we can study it once a month, right? So or even if there are other groups that have spawning once a week because they have multiple cultures. So we can really learn things rapidly with Aptasia. So then when we look at what's happening then as the algae enter that gastric cavity, you can see that they are engulfed by a process called phagocytosis. And you can see here the pseudopods, oops, sorry. How do I go back? Oh, it doesn't matter. The pseudopods reaching up and engulfing this alga here. And the process is highly conserved, and it's essentially identical to the process that happens in our professional phagocytes in our immune system in our blood. So phagocytosis has been around forever, amoebae to humans, and those pathways, now that we have genomes, we can see that those pathways are very highly conserved, and it's those pathways that are engulfing those algae in corals. But so once they get engulfed, they end up in the gastrodermis, but crucially, they persist, okay? They do not get digested. They do not break down if it's the right type of alga, and that's a whole other story. But they persist. And so what are the processes that lead to engulfment and persistence of algae? How does that happen? It's a cessation of immunity somehow. So now let's get down and dirty here. Okay, so phagocytosis is complicated. Like I said, it's highly conserved, it's canonical. So you can really look for the same things across many taxa. But to start simply, like in a professional phagocyte, you have a microbe that has these decorations called microbe-associated molecular patterns that hang off of it, that it sometimes can't control. The good parasites can control them, but the typical microbe may not be able to control what's hanging off of them. The host is set up to receive and detect those MANTs using pattern recognition receptors. And once that lock and key thing happens, it can launch a variety of signaling cascades that can drive destruction of that microbe if it's a negative invader. So phagosomal maturation, microbial killing mechanism, inflammatory response, apoptosis, autophagy, lots of different pathways. So here we are with the Symbiodiniaceae being the microbe, and we have now a host cell. And I've then arrayed here some of the different types of signaling cascades, MAMPs, and lectins, or pattern recognition receptors that are known across, biology. And you can ask, which ones of those are in our system. And so the Symbiodiniaceae comes in contact with a host cell, and in a healthy partnership, the alga persists, and that symbiosome gets altered. And we now know that it's kind of a phagolysosome altering, but that's another story. That's not my story. And somehow these processes get blocked, okay? And the symbiont takes up residence in the host cell. How do we know what those genes and proteins are? Well, we have to discover them. And we discover them using a sort of a a variety of omics and high-throughput sequencing approaches that are now really standard, but there's been a cascade of them. And I like to say, back in the old days, when I was actually pipetting, I actually did this stuff so long ago that it wasn't even called omics. It was just 2D gels. But now it's proteomics. So, I did proteomics. But today, there are lots of very cutting edge, really exciting tools. We saw lots of these yesterday in the talks. One thing that's not even up here is there's now spatial transcriptomics, so you can array and look at where transcripts are on a microscope image. It's nuts. It's very cool. But lots and lots of people are doing this work, including folks in my lab. So many published omics studies now, including from my group. So these are photos of some of my current and former students and postdocs who've done these kinds of work. So their goal has been to explore the role of these candidates that they identify using these omics tools in regulation. And so here I show just all of the people that I can have in my mind that have studied these pathways. Check mark is a publication or more. The blue people are my current students and postdocs. And actually today, just after the coffee break, Olivia is speaking at 10:15 on the SMAD ChIP-seq project that she's performing for her thesis. And then right after that, June Chai is going to talk about sphingolipids. So I hope you'll come to those. And I also just want to point out that this kind of work is also leading to some solutions-based projects. We have a new project, Daniel Wang-Prasert and I, from Revive and Restore, where we're going to be examining the role of glycans in solutions-based resilience for corals. So in a healthy partnership, as I said, the alga persists in the symbiosome. But during dysbiosis and bleaching, we know a lot actually, but it's puzzle pieces, that the innate immune mechanisms that have been blocked are re-engaged. And I want to pause for a moment and just talk a little bit about bleaching. And sort of, we can ask this question, what stress response does this trigger cause? So let's talk about heat stress. There are lots of stresses, but we'll talk about heat stress. What stress response does this trigger cause? And we really can put the stress responses into two categories. The first is on your right, oxidative stress of symbionts. This is a concept that's been, actually Michael Lesser was a big early researcher in this area. There are hundreds of papers on oxidative stress in corals, but what we know is that with elevated temperature, heat stress causes stress in the algae. The photosynthetic apparatus has a kind of an Achilles heel part of it, and that becomes destabilized. The oxidative stress then causes damage in the algae, and those reactive oxygen species can move into the host. It's toxic, and the algae are lost. But crucially, that launches a huge immune response in the host. It's a huge, complex immune response. It's like oxidative stress. You don't want oxidative stress in your cells, like, get this thing out of me, right? Okay. So then, but a newer and equally compelling, and not if, you know, it's not one or the other, it's both of these things are likely happening depending on the situation. This concept of metabolic dysregulation, where here we have the healthy cell that we talked about earlier, but under heat stress, you can see that arrow from the host gets big, and the host loses control of its metabolic stuff, and it starts to catabolize its own proteins because it's elevated temperature and it's stressed. And that allows the symbionts to say, hey, you know, I don't need to send this carbon to you anymore. I'm going to keep it for myself. So the photosynthase stops moving to the host, and the symbionts move towards parasitism, and the host moves towards starvation. and the host says, get this thing out of me, and the algae are lost. How does the host, how does the symbiosis break down? We know that there are a variety of ways. There's in situ degradation, where the algae are destroyed. There's exocytosis, where the algae are spit out. There's a high stress response, where the whole host cell just detaches from the membrane. There's host cell apoptosis, so the host cell kills itself. The host cell dies by necrosis. There's autophagy, which I don't show here. So the endpoints are complex, but they're all really coming from a variety of immune responses. But crucially, again, from a solutions-based point, coral holobionts are differentially susceptible to bleaching. So this slide shows that in the same environment, different coral types can be differentially susceptible. And that is another huge area of interest that's driving many of the talks this week, trying to understand how some are resilient and how others aren't, and how to breed that, or how to understand how we can help with driving resilience. So I want to tell my first little story today about some work that my former student, now PhD, Val Sawicki, examined on phagosomal maturation and microbial killing with the NOx protein. So this is my first little story. And let me first introduce you to NADPH oxidase. It is a classical part of an innate immune response. It is a multimeric protein enzyme that assembles itself on the phagosome membrane and produces an oxidative burst, so makes superoxide inside of the phagosome and essentially digests a foreign invader. And so if a foreign invader comes in, the multimer then comes together, assembles itself, and then produces these superoxide radicals and destroys the invader. And we know from our omics work, my group, but other groups as well, have picked out NOx as being differentially expressed as a function of symbiosis and as a function of bleaching. And so we can ask questions. So we want to know what the function is. We've described, the omics is great for hypotheses, but we now want to know what the role is. And so Val posed some questions. And she asked, is NOx repressed in the presence of algae? And so you can do this with Aptasia because you can compare aposymbiotic animals versus symbiotic animals. And she proposed that in the presence of algae, that NOx would be blocked and that phagosomal maturation would not proceed. And so that would lead to persistence of the algae in the symbiosome. And she measured expression levels of NOx and protein levels. So I'll show you those data in a minute. And then she was also interested in NOx in the process of heat stress. Is NOx activated with elevated temperature? So she proposed that with elevated temperature, NOx would be turned on by the presence of these sick algae, right? And that it would drive the cell towards phagosomal maturation. And she used expression levels, protein levels, activity and localization to study these things. So let's look at these data. So first, this is in the aposymbiotic versus symbiotic animals. She showed that NOx and gene expression are repressed in symbiotic animals. So if you compare symbiotic versus aposymbiotic animal qPCR expression, any down bar is showing, it's hard to keep track, any down bar is showing a repression in the symbiotic state. She showed that 3 of the subunits were significantly repressed in the symbiotic compared to the aposymbiotic state. And when she made antibodies against 2 of those subunits, she was able to show in the aposymbiotic state, it doesn't matter. In the aposymbiotic state, they were higher than in the symbiotic state. So in symbiosis, NOx is quiescent. So all is well, you don't need to destroy me, right? And so she was able to show that. She could show that the phagosomal maturation was blocked by the decrease in NOx. So repression of NOx is consistent with blocking of phagosomal maturation and symbiont persistence in the symbiosome. And then she moved on to asking the same sets of questions in a stressed host. So she found that both NOx gene and protein expression increase with heat stress. So here she's showing with her qPCR a modest increase in these two, no significance there, but a modest increase in these two subunits, NOx2 and P22-FOX. And she showed a modest increase in protein expression of these two genes, or proteins. And then she did a NOx activity assay using cell rocks, which is a fluorescence protein, and that should show reactive oxygen in an in vitro situation. And she was able to show that when she added sample, with heat stress, she got a huge response, a huge reactive oxygen response that she could abolish by adding a scavenger, a Ross scavenger. And she could decrease by about 40% or 60% when she added a specific NOx inhibitor, which suggests that this increase in Ross is due to the presence of NOx. She also did a beautiful immunofluorescence, epifluorescence, confocal microscopy, and she was able to show a migration of signal, an increase in signal, and a migration of signal to around the symbiosomes in 5 days of heat stress. So these are aposymbiotic animals. This is symbiotic animals at the start of heat stress and then after 5 days, and you can see in 2 different antibodies this migration of signal to around the symbiosome. So she concluded then that NOx activity and location of NOx is consistent with phagosomal maturation and bleaching. So this is just one little story of many stories I could tell about the immunity kinds of questions that we ask in my lab. So to conclude this part of the talk regarding immunity, in health, when the innate immune system is balanced, when the host is tolerating the appropriate symbionts and resisting perhaps diseased things that could cause disease, there is a healthy interpartner communication. The partners are avoiding immune destruction of the symbiont, and there's a dynamic homeostasis. And then in contrast, during bleaching, the teeter-totter moves towards resistance, and the host wakes up. It's like my analogy is always Harry Potter's invisibility cloak comes off. Get these things out of me. They're toxic. There's a communication breakdown. The holobiont becomes stressed, and the corals lose their algae, and symbiosis moves to dysbiosis. Okay, the last little part of my talk, I'll talk about host-symbiont biomass regulation. And this is another area that the Weiss Lab has spent lots of time through the year studying. And we can ask questions about how do the partners regulate their numbers together? How are symbiont populations regulated during onset and maintenance of the symbiosis? How do symbionts invade growing host tissue? How does host nutritional state affect symbiotic development and growth? So lots of questions, again, that are 50 years old, but are still very relevant today. So just to push on the model system a little bit, just to illustrate this a little bit more, just a couple of examples. So here we have an image of a Fungioscuteria larva that has a few algae in it. So you can ask, how does a symbiont population keep pace with host growth? So as that little larva goes through settlement and metamorphosis and starts to grow, how does the algal population regulate itself? Another example is, many of you are familiar with acroporids having those white growing tips. They're non-symbiotic. Well, how are the algae invading that? And how is that regulated? And once again, Aptasia is an incredibly powerful model for understanding these, or trying to study these kinds of questions. in large part because it has both sexual and asexual reproduction that we can now study in the lab. So it has a typical eggs and sperm released, creating a planula larva, and then onset of symbiosis can occur either during the larva or as a juvenile. But also, the algae, or the host can undergo pedal laceration and just walk off little bits of foot that then undergo a developmental transformation, a morphogenesis, and make a new polyp. And so you have both of those types of life histories that you can study in the lab. And once again, you can also make apos. So those aposymbiotic animals are powerful because you can study, you can compare and contrast between symbiotic and aposymbiotic. So again, another Weiss Lab hangout is this study of biomass regulation, both in adults and through development. And through the years, we've asked these questions as a function of symbiont type, as a function of temperature, as a function of feeding. We've done a variety of studies just in adult aptasia where we've looked at onset of symbiosis and symbiotic maintenance through time. A lot of this work was done with Simon Davies. So Simon Davies' lab pioneered a lot of this that we collaborated with. So Yasmin and Lucy and my student Trevor have collaborated on these questions through the years. A lot of publications on cell cycle control coming from Simon's group that has really shown how the host and symbiont are regulating these processes. And again, as a function of nutrition and temperature. And then in aptasia during asexual development, what's the timing of development in the symbiotic compared to an inoculating animal, and as a function of nutrition, as a function of temperature. And then many, many students of mine through the years have studied these processes in larval corals. Questions about specificity, so different types of symbionts, and with heat. And so I just want to point out that my former student, Dr. Eric White, is giving a talk about his work on nutrition in aptasia on Friday at 9:30. So I want to tell my final little story about host nutritional state and how it affects symbiosis, or host development and growth. And this was work that was started by a developmental biologist postdoc in my lab, Jason Presnell, who was able to show that the progression of symbiosis, first of all, he described this progression of morphogenesis, and then he asked whether or not the symbiotic state made a difference in how fast things happened. And he was able to show very early on that being symbiotic was a good thing, that you grew faster. And so another postdoc of mine, Dr. Sam Bedgood, came along and he said, well, you know, I'd like to extend that timeline out, and I want to overlay this question of nutrition, because Jason's work in the early days was all just starved animals. And so Sam's hypothesis was the presence of symbionts in pedal lacerates will boost partnership growth and survival during starvation. And he did this experiment, again, because of the power of models. He used 3 different types of manipulations. First, he did aposymbiotic animals throughout. Then he inoculated a series of animals after they developed a mouth as pedal lacerates, and then in symbiotic animals. And he either starved them, didn't feed them at all, or fed them little itty-bitty rotifers, because the pedal lacerates are too small to feed brine shrimp. And he followed them for 60 days, so it was a little bit of a longer experiment. And his results were shocking. I was shocked. It blew all of the whole symbiosis is great out of the water. So the lacerates, first of all, if you were fed, you did great. Even the aposymbiotic animals, they did great as they were fed. So any filled circle is fed. But if they were starved, the apos still did fine, the aposymbiotic animals did fine, but the symbiotic animals and the inoculated animals, frankly, they died. Okay, so they couldn't take that starvation. and the symbiosis went to hell. This was shown also by the growth of the animal. Once again, everybody who was fed grew. The aposymbiotic animals that were not fed, they just stayed put. They didn't do anything. They didn't grow and they didn't shrink. But the symbiotic animals and the inoculated animals shrank. They just got smaller and smaller. And so crucially, growth decreased in symbiotic animals, and symbiont population size increased. So the symbionts were having a grand old time. So the population size of the symbionts in starved animals increased, which suggests that the symbiosis had moved towards parasitism. So this was a really surprising result. The idea that symbiosis could get you going in life as you're getting started really kind of fell down, and that the role of heterotrophy became incredibly important. So starvation in juvenile causes nutritional dysregulation, homeostasis breakdown, and the algae become parasitic. So we can go back to this slide about coral bleaching, what are the responses, and we can change it a little bit. And we can say metabolic dysregulation, bleaching versus starvation. Well, we already learned about metabolic dysregulation with bleaching here, and now we can add on what we've learned from Sam's work, and we can say that metabolic dysregulation in juveniles, when you withhold food, is that starvation causes changes in nutritional regulation. Maybe the symbionts move towards parasitism. Perhaps photosynthate is no longer moving. We haven't tested these things. But the algae persist, and that's really interesting. Is there a developmental component? Is the immune response blocked? So that's, I think, very, very interesting. And so we don't have bleaching. Host nutrition depends on developmental stage. Is there an immaturity to the immune response in these juveniles that we need to understand? Could this have implications for fragmenting corals and perhaps stressing them with no feeding early on? Ripe for future research. So to conclude this section then, in health, nutrient cycling in partners is regulated, host size and algal population size is balanced, there's a dynamic homeostasis. But in sickness, nutritional dysregulation happens, symbionts move towards parasitism, homeostasis is lost, and the immune response, at least in juveniles, seems to be repressed. So to conclude, all of us here are working to preserve reefs, and to help with solutions to the coral crisis, we cell biologists are focusing on the future. We're adopting solutions-based approaches in our work in various ways. And we're emphasizing cooperativity and rapid action by studying models, by sharing information. And Gandalf says, very appropriate for New Zealand, not even the wise know all ends. We don't know how this is going to happen. We can't give up. We have to keep moving forward. And with that, I'll just say this is my lab right now. Livia and June are here. Eric is here. He's a doctor now, so he's still a Weiss Lab member, but he's recently graduated. And then Maria's not here because her due date for her first baby was yesterday. And we haven't heard anything, so I guess she's still pregnant. So we're hoping she's doing great. And then my collaborators through the years, Sandra Luskin and I have been working for years on glycans. Nate Kirk and I at OSU have done amazing educational work with aptasia. We have 1,200 students in introductory biology studying aptasia. And then, of course, my colleagues Simon and Arthur, who's not here, we've been working together for years. I've been getting to come to New Zealand. I think it's my 7th trip. So thank you, thank you, thank you for inviting me, and thanks for your attention.
I'm sorry to say we don't have time for questions. You can catch Virginia during the meeting. But the Society wanted to give you a little token of their appreciation for being here and giving us a talk.
Shall I open it in front of all of you? No.
Thank you.